Very good morning to all of you. So today I'll be talking about one patient who was just admitted with us last week, and the interesting insights that we got from trying to understand his clinical phenomenology. So the title is called Self-Motion Induced Kinetopsia and a Popout Illusion. And as we go, we'll see. So in this slide, you can see. So I want you to imagine that you're going on a bike ride along the countryside, and so the wind is on your face, and you're rushing up at high speed, and you realize that as you go fast, all the trees are whizzing past. The road sides of the roads are all going radially on either side, and suddenly this little boy being chased by a dog comes across, and very effortlessly we are able to press the brakes and stop. So if you try to stop for a moment and try to ponder how this complex process happens, it's quite amazing and it causes wonder because so you have to integrate so much of information. So as you move forward, all the objects are darting across your retina, and versus objects that are actually moving are also darting across the retina. Your brain has to integrate all of these confusing information. So like whether the tree moves because you're moving or whether the boy is crossing the road, the retina will not be able to differentiate. It's only that the brain that has to integrate all of these differing information and come to a very stable percept. So as you move, you realize that the objects that you see in the environment are darting across on either side, and that is called as optic flow. So optic flow is a very important perception because it tells you whether that the direction in which you're moving. It also tells you that what kind of movement. For example, if you're moving forward, the optic flow would be radial, so things will be moving on either side. If you're moving your head side to side or you're doing pursuit movements, then the movement will be planar. And if you're moving your head round, then the optic flow will be spiral. And so optic flow is used by the brain to try to interpret the direction in which you are heading. And the problem is that optic flow will also be due to objects that are really moving. So when an object moves, that also causes the object to dart across the retina. And it's important for the brain to differentiate the optic flow caused by self motion versus the optic flow caused by objects in motion. And so it was interesting because all of this we had no understanding of till we came across this patient. This was a 31-year-old gentleman who had come to us, was admitted with us with a history of headache, seizures, followed by altered consciousness, and it subsequently improved. And we had diagnosed a venous infarct involving the occipital lobe. And when we spoke to him, we found that he was riding his motorbike home, and suddenly he noticed that severe onset of headache. And this caused him to stop riding his motorcycle, and he stopped. And after that, when he tried starting his bike and moving forward, every time he moved forward, the object in the background, which is in this case the tree, and in his phenomenology was a temple and a building, moved towards the center and almost collided close to the midline. So he was extremely disturbed by it and he stopped. And when he stopped, then the building and the trees and all moved back to the original space. And this happened about 3 times. Somehow he managed to reach home. He continued to have severe headache, so he decided to watch TV along with his family members. And when he watched TV, he noticed that objects in the TV were moving abnormally. So it looked as if they were shaking when they were not actually shaking. They were all a bunch of people who were standing and talking on TV in a serial, and it felt that they were all shaking along the horizontal plane. So this was the phenomenology that he had presented to us, and this got us thinking into why this really happened. So one thing we could understand is that there's something to do with optic flow, because every time he moved, he thought that the environment also was moving in the opposite direction. So normally you would expect the environment to move, but it will move radially so that you know that you are moving forward. But here it was moving in the opposite direction. So we realized there's something wrong to do with optic flow. So this is the clinical scan that was done for the patient. So you can see that in the right inferior occipital area there is and corroborated by the filling defect. So this set about us going to understand this phenomenology. So we wanted to understand what are the brain areas that process optic flow. So we did a lot of reading and we found that there are multiple fMRI studies on this and a lot of monkey recording studies as well. So most of these studies are done with either with a human being in which they are showing optic flows. Optic flows are recreated by actually showing multiple dots moving in a particular direction. So it's like a lot of dots moving away in one particular direction may create a illusion of an radial optic flow. So it's like as if you're moving forward, versus you can have dots moving in various directions, creating different kinds. So there are a lot of standardized fMRI experiments, and then they found that certain areas are active in this. The most common is the human area. The motion area is area MT and MST. In human, these two form a cluster, and so that's why they are both seen in red and green together, and they are sensitive to optic flow, and also for movement of objects as well. I would like to highlight there are two areas, V3A and V6, which are again part of the dorsal pathway of visual perception. They are involved in kind of recognizing selective only to object motion, and V3A is selective for head, and it's not affected by eye movements at all. And as you go anterior to the parietal lobe, there are two. One is the cingulate sulcus area and the intraparietal sulcus. These are quite important in integrating it with vestibular information, because vestibular information gives a lot of valuable information about eye position, head position, etc. So all of these get integrated. So this is what we understood. But one thing when it comes to cognitive clinical phenomenology is that how do you know that the patient is just not making it up? So one thing that we always want to do is kind of objectively have some test to prove that this particular person has this deficit. So we did a bedside clinical assessment of his visual function, and we found that he has a small left-sided hemianopia. Some color perception he was making small errors in color naming, but otherwise he had no major deficits at the bedside. So we decided to study in the lab. So one way that motion processing in the brain is studied is using a particular test called as the motion direction discrimination task. Here, what we do is we do a lot of random dots are taken in the central visual field, and all of them move either in one particular direction together, and then they vanish and they keep. So individually the dots in a second, but as a group you will realize that all the objects are moving towards one particular direction. So this is a standard test, and so we decided to do this test on him, and we found that this is completely normal in him, as what we'd expect in normal people. So, but our hypothesis was that somehow when you bring in optic flow, this thing would get disrupted. So another way to study movement perception in little more detail is called as a motion coherence task. So here only a proportion of the dots move in one particular direction. So the dots can be completely random in the sense that they are not having any particular direction, or only about proportion of them, either 10%, 20%, up to 100% will move in one particular direction, and these will be presented randomly. So and using this, we'll be able to guess what threshold each person has. So normally the threshold varies. We've found that the threshold ranges somewhere between 20% to 90 to 100%. People with migraine especially have a higher thresholds for motion perception. So we decided to do this on him, and then when we did this, again we found this was quite normal, and for him we found that the threshold was 40%. So this is a scatter plot showing the results of the patient for the motion coherence task. So here 0 stands for all dots are random. When all dots are random, you can find that he is accurate, and above 50% also he is accurate. So if you can see that the pink triangles are the ones that are all the errors. If you see, all the errors are between 10% and 30%. So we calculated that his threshold or motion coherence threshold is at 40%. So now we had to actually prove that this gentleman, when you introduce optic flow, has a problem with visual perception. So we decided to introduce optic flow. So we had to design a specific task for this, and we wanted to use a motion coherence that is above his threshold. So we decided to use 50% and 100%. So this is a design of the task that we used for him. So this particular center is actually, we've colored it here so that we can see what is the field in which the dots, the target dots will move. But during the test, actually this colored disk will not be seen. So in the center, we do the same motion coherence task where the direction, you have to guess the direction, and only 50% of the dots will be moving in one direction or the other. And but this is actually there are a lot of distractors all around. So we have around 2,000 dots moving in very fast and all radially away from this. So this produces an illusion of optic flow, or the person is moving forward. And so this is what we did on the patient. So we also want to also affect him. So we moved his position while he was doing the test. We manually rotated rotated his head to stimulate the vestibular pathway and cause pursuit or oculocephalic movements. And we did this test in different conditions. So, and these are the results. So, and another component of this task is that the motion sensitive areas are extremely sensitive to contrast. So, like white versus dark. So, white, some areas are more sensitive to low contrast, which is white, and some areas are more sensitive to black, which is a higher contrast. So here, when we did white dots with white background, background dots are also white, and all the dots were 100% moving in one direction. He got everything right. So, but the moment when we introduced 50% coherence, we realized that he started making 60% errors. So before we did the test on him, we did a group of controls with, and we found that almost everybody got everything correct in this task. So this is definitely an abnormality in him. And when we made it into black dots, he was a little better. And black dots in a white background, again, he was a little better. So here you can almost get like a dose response kind of pattern. And we were able to demonstrate what he experienced subjectively could be objectively demonstrated in the lab. So introduction of optic flow produced difficulty in direction discrimination perception. So we did a lot of other tasks. So we also found that he had some errors in color naming. Another interesting problem that he had was he had a hemimacropsia. So objects shown on one side of the screen, he always viewed slightly bigger, and he was able to recognize emotions. He was able to recognize faces, and we also did the same task with head movement. So we were moving his head, and he was still able to get the moment perception correct. So now we objectively proved that this gentleman has got subjective visual phenomena, which is called that as he moves forward, there's an environmental illusion of movement. So we decided to call this self-motion induced environmental kinetopsia. So now we wanted to go ahead and try to do some neuro imaging. So this is a clinical MRI. So what we did is then we manually converted this lesion location into the standard MRI template, which is called as the MNI template. And this will tell us which all areas in the brain are involved, because you can overlay the Brodmann's atlas and you can overlay fMRI studies. And not only that, we did a resting state fMRI on this patient. So resting state fMRI is the patient is asked to lie down with eyes closed, and bold scans are taken over a period of 5 to 6 minutes. So you have 300 scans or 300 data points for each voxel. And what you can do is, and you can do a correlation coefficient between each of these voxels to all the which areas in the brain correlate with all the other areas, and this will tell us about the resting state connectivity. Another interesting thing that can be done is that you can take this particular area of lesion and we can put it in a large data set of normal resting state fMRIs and see what this area is connected to significantly. So this is called as a lesion network. So lesion network will tell you that sometimes even if the lesion does not explain the deficit, the lesion network, because by virtue of it being connected to other areas, will tell you what the deficits are. So that's what we did for him. And also another thing is that so we had to come up with a model of how optic flow is involved. So we did a lot of reading and we pulled out all the literature that processed all of these fMRI studies, and we found that these are the areas. So we postulated that V3 and V6 are important areas because V1 and V3 are non-selective areas for motion perception. They kind of respond to almost all kinds of motion. And area MT and MST are responsive to both, but it's a higher level area than V3 and V6. So V3 and V6 will probably tell area MT and MST then whether the movement is self-induced or whether it's caused by something else. And subsequently, downstream areas such as the CSV and IPS motor are like perceiving which direction you are heading. So if you have a problem in V3 and V6, it's likely that you may end up having a problem with optic flow, separating the optic flow from object motion. So we kind of built this model, and the other thing that we had to do is, because we had to do an fMRI analysis, so we had to pull out the coordinates of all the fMRI data, and we had to create what is called as a region of interest. So we generated a 6 millimeter sphere for all of these areas, location in space, and then we wanted to look at the fMRI data from those regions of interest, and we did a correlation analysis. So what we call this is the optic flow network, and then we looked at that particular network using our patient's fMRI data. So we had a group of 5 normal controls taken from the human connectome project, and then we've taken R-bound patients resting state fMRI, and we looked at these nodes that are involved here, and we did a lot of using the general linear model of analysis. We found that these are the nodes that are different. So the way to interpret this is that there are various there's a method called as graph theory that analyzes brain connectivity, and all the areas that you see in red are the areas that are different in the patient group compared to the control group. So what we have demonstrated is that this particular network that is abnormal in our patient compared to normal groups. But how is it useful clinically? So we wanted to understand whether these two particular areas are involved. So what we did is that this is a simpler method where you just have to overlap networks. So we took the patient lesion network and mapped out the other two areas, which is area V3 and V6, which we postulated in our hypothesis that may be involved in this deficit. And here we were able to demonstrate that the lesion was actually overlapping significantly. Lesion network was overlapping with V3 and V6 areas. Then proving our hypothesis that this patient is having problems in processing optic flow because of the lesion network affecting V3A and V6 areas. So we thought all of it is over, and then so while we were reading up, we also read we found something on visual illusions, and we found that most normal people, when they see an illusion like this, will see rotatory movement. So we had a task in which we were showing the patient all these rotatory motion, these illusions, and we found that the patient could not see rotatory movement at all. Instead, that he felt that the entire thing was popping out of the screen. So that is another interesting phenomenon. And actually, previously, a few years ago, we had a patient like that with posterior cortical atrophy. So he was watching TV. He used to get scared that the characters in the TV were jumping out of the screen. And there's a lot of gaming literature saying that if you have a screen or a frame within, if you frame a picture that appears it's coming out, then that will be exaggerated. So we decided to actually study that a little bit more. So we designed a task again, so it was quite easy. So we pulled out some images. So we got help from everybody in the team to get images, so the patient and controls had to say whether the object was appeared jumping out or not. So we had two conditions: whether the object was framed and whether the object was not framed. So there was some gaming literature that said that if you frame, then the 3D effect is more and it looks like the object is jumping out more. So, and when we did this task, we found that this is the spread of results. For controls, most people, for some of the pictures, some normal people also felt that it was popping out, and that's what gaming literature also said. And but for the patient, we found that this was significantly much more. So we had actually made a sliding scale on the Likert scale, and then we did about 15 to 20 controls, and we did the patient, and we found that there was a significant difference in the scores between the patient and the control. So, and this is how we were able to again objectively estimate what he. So then we went back and read. So this is again a publication from our department when we looked at when we had described popout illusion, and we went back and read the paper and to find out what is the exact cause, and we found that there is something called as figure and ground. So whatever comes in front is called the figure, and the background is called as the ground. And there are certain areas in the brain that process figure ground segregation, and this is also an important part that processes frames of reference. For example, a television screen is a frame of reference. All objects inside that will remain inside that. And similarly, a door or something like that. So you don't expect things to move in and out of a frame of reference. So, and we found that this retrosplenial cortex is an important node in processing environmental frames of reference. So we figured that this patient probably has a problem in the retrosplenial, or the lesion is extending into the retrosplenial cortex. So he has this kind of problems. So now, if you go back and see that, so a lot of motion perception actually requires frames of reference. So you need an environmental frame of reference, then you need a head frame of reference, you need a retinal frame of reference, and each object have their own allocentric frames of reference if you want to actually understand. And if you see neuroscience literature, there is, especially if you search retrosplenial cortex, there's an entire group where they study navigation. So navigation was studied mainly with hippocampus and the parahippocampal gyrus and the lingual gyrus, and people have studied using maps and direct brain recordings, but nobody's actually studied optic flow in them. And we figured that both processes have to be connected, and there's hardly any connect in the literature, because people who study optic flow are giving visual stimuli and recording from the brain, while people who are using navigation are using either stimulations or they're studying map navigation. And coming to, if you think about it, both processes had to be linked, and but we find that there's hardly any linking literature on that. And but one thing that we did find is that retrosplenial cortex and its connections to the visual cortex is being mentioned, but not many studies are related both. So then we came across a big gap in literature saying that there's an entire study on optic flow that so all of the meta-analysis of all of these results, they don't mention retrosplenial cortex or frames of reference, and all the guys who talk about navigation don't really talk about optic flow, except for one which I'll come to. So we decided that whether will we be able to bridge the gap. So what we did is we built a model that included all the other areas, we included all the areas, hippocampus, we included the retrosplenial cortex, and we decided to study the fMRI in our patient to see whether it's different. And what you can see here is that in our patient, the retrosplenial cortex, which is over here, is showing strong connectivity with area V6 and V3, and this connectivity is definitely different from that of the normal controls. So to explain it simpler, so this is a lesion of the patient, and what is yellow is the retrosplenial cortex, and the lesion network is the connections of the lesion area. And we were able to demonstrate that the lesion is connected to both the retrosplenial cortex and V6. So, and we were able to demonstrate that a single lesion, which is involving probably V3, V4, and V2, is disrupting both networks. And we also want to postulate that both these networks are involved in similar processes, which is processing self motion. And subsequently, we did find one research that says that some navigation ability and optic flow areas are very similar, and they also have come up with something, a similar model to what we had used. So in conclusion, so I would like to say that interpretation of self motion induced optic flow and perception of frames of network are interlinked neural processes, and because the methodology is different for both, basic neuroscience research, actually there seems to be a gap, but nobody talks about it, which I'd like to call it as a silent gap. And studies like this, especially, can bridge this gap. So here we have demonstrated in one single patient that lesion affecting one process can also affect the other. And also, I'd want to highlight that we should be open to novel clinical phenomenology, and because this is what makes modern clinical neuroscience relevant, because in this patient we were able to do rapid hypothesis generation, we were able to read up, build models, test it, and come to our own conclusions using contemporary neuroscience methodology. And this is what makes lesion based studies, which was quite prominent in the early eras of neuroscience, still relevant in the modern era. Thank you. So he came for a follow-up. So a lot of these are, so some of